All right, let's go ahead and get started. So thank you so much for joining us today. Let's welcome Professor Fekrije Selimi. So she has served as research director of the Center for Interdisciplinary Research in Biology at the Collège de France since 2011. There, she is leveraging the model system of the olivocerebellar network to pioneer new approaches to uncover and characterize molecular pathways that control synapse formation and plasticity of neural networks in the mammalian brain, especially in the context of motor neurodevelopmental and cognitive disorders. Prior to starting her lab, she trained in Paris, attending École Normale Supérieure, and earning a master's degree in biology and biochemistry in 1996. She went on to earn her doctorate in neuroscience from University Pierre and Marie Curie in Paris in the laboratory of Professor Jean Mariani in 2000, where she researched molecular mechanisms of neurodegeneration in the Lurcher mutant mouse model, and then was an HFSP postdoctoral fellow in the laboratory of Professor Nathaniel Heinz at the Rockefeller University, New York, where she investigated synaptic diversity in the mammalian brain and performed the first purification of a single synapse type from the mouse brain. She is the recipient of the Boehringer Ingelheim Fens Award in 2010, a Tabavinier grant in 2011, and an ERC Consolidator grant in 2016. In addition, she serves as president of the France Cerebellum Club. We are thrilled to hear from you today, Fekrije.
Hi. Yeah, thank you very much for the invitation. So I'll start and share maybe my screen. And thank you all for attending. I see some friends' names on the list. It's nice to see you all. So I'll share my desk. You all see it? Yeah?
Yes.
Okay.
Yes, your slides are here.
Yeah, great. So, what I'm going to tell you about today is the work of my team that we've been doing in the past few years. And first, I want to add that I'm also an ALBA ambassador. So for those of you who might not know it, the ALBA network works towards diversity and equity in brain sciences. And it's very important for me to mention that and to also show you on this slide the network and websites where you can find all sorts of informations that are important in this regard. So my lab is interested in what's happening in the brain of mammals at a stage development where basically the brain is a synapse factory. And for humans, the peak of synapse density is achieved after birth. But as you know, acquiring the capabilities of the mammalian brain that are huge in terms of computation in particular, and all the cognitive functions that it can do is not just a question of making a lot of synapses, but of making the right synapses. And you have a lot of diversity in terms of synapse formation in the brain. And here I'm illustrating the diversity of synapses in terms of their morphology. So I think you see my pointer here, where in pink you have presynaptic boutons contacting different types of postsynaptic domains in blue. And this type of diverse morphologies of synapses is accompanied by diverse functions that are essential for a proper circuit function. So you have to make synapses, diverse synapses, and you have to make them precisely so that you generate stereotyped circuits and stereotyped patterns of connectivity. And in particular, we can find patterns of connectivity that share similarities across the brain. And here I'm showing three types of neurons in the brain, one in the cerebellum, we'll talk about it much more afterwards, but one also in the somatosensory thalamus, and one in the dorsal cochlear nucleus. And in these neurons, they receive several types of glutamatergic inputs that make synapses that are different for many types of reasons. And one of the reasons is where they connect the target neuron. So in orange, you see that some fibers will connect the proximal dendrites of a target neuron. And here you can see it for the Purkinje cells in the cerebellum, here for neurons in the thalamus. And here you see in the dorsal cochlear nucleus, these are not proximals, but a specific part of the dendrite in these neurons. But these three orange labeled inputs, they bring an organized information. So in the cerebellum, it's a topographic organization of the climbing fibers. In the thalamus, it's somatotopic organization of the lemniscal fibers connecting the target neuron. And in the dorsal cochlear nucleus, it's tonotopic organization. On the other hand, these three different types of neurons also receive other glutamatergic synapses that bring multimodal information. And those connect different territories on the target neuron. So how do you reach this very stereotypic and precise type of connectivity in the brain? How do you develop this? We've reduced the question to what controls synapse identity. And what I mean by that is that each target neuron, each neuron in the brain will receive different types of inputs from different types of neurons that will make synapses on their target with a specific subcellular territory with specific numbers and will have specific functional properties. And this is what I call synapse identity. This is not a trivial question because neurons just want to make synapses. So here I'm giving you two examples to illustrate this point. But so eutaptic synapses have been described pretty early on and have been shown to be basically the ability of a neuron to make synapses on itself if it doesn't have the right partner. And here, this example is the example of a neuron cultured in isolation on a bed of glia. And here, stained with synapsin, you see that the neuron makes synapses on itself. Here, in a study on a mutant mouse that shows degeneration of Purkinje cells in the cerebellum, we can see that in a model where we preserve the inputs, the granule cells to those Purkinje cells, in the absence of their target, they try to make these granule cells make synapses on glia. So really, it seems to be an interesting intrinsic property of neurons to make synapses. So how do you control the proper formation of precise and diverse synapses during development? And along in the 60s, Perry proposed the chemoaffinity hypothesis that basically suggests that for each synapse type in the brain and on a given neuron, you'll have a specific combination of molecules that will instruct proper synapse formation. Up to now, however, this hypothesis hadn't been really tested. And this is one part of our work, which has been to test this hypothesis directly to determine whether indeed each synapse type is defined by a specific molecular composition. If this is so, what is this molecular combination made of? And if we find this molecular combination, is it really instructive during development? So I'll show you what we have done on this subject in the rest of my talk. But another hypothesis, which was postulated in the 70s in particular, stated that basically the selectivity of synapse formation and the preciseness of circuits in terms of their synaptic connectivity was due to the selective stabilization or elimination of synapses during development in an activity-dependent manner. And I'll show you how we also try to understand whether these two hypotheses that seem to be opposite at this time might actually be interconnected. And you'll see at the end of my talk why I'm saying this. So addressing this question is not trivial because, as you know, even in a very small volume of the brain, there's a huge number of synapses. And along the years, thousands of different synaptic proteins have been identified. So how do you determine what is where for specific synapse types? And so over the years, we have developed a strategy in the team using cerebellar Purkinje cells as a model, but the idea was to identify the protein composition of specific synapse type on a given neuron, both presynaptically and postsynaptically. And for this, we developed techniques that allowed us, using genetically modified mice, to do either synapse-specific proteomics or neuron-specific transcriptomics. And once we identified this protein composition of specific synapses, the idea was then to change the synaptic code, if you wish, in the mouse during development, either by removing some proteins from where they should be or misexpressing them where they shouldn't be, and then analyzing the morphological and functional consequences of the change in the molecular synaptic code during development. So with this audience, I don't have to go into details as to why it's interesting to study the cerebellum. I'm not going to, go on and read Jeremy Schmann's citation. But for us, actually, it's not only important to study the cerebellum because of its potential role and what interesting new information we can get about its role, but for us, one of the key point in using the cerebellar cortex as a model in particular is that we know very well the connectivity in this structure. Since already Ramon y Cajal, we knew all, maybe not all, but the major cell types were already identified by Ramon y Cajal during his studies, and we knew how those different cell types are connected. And in particular, cerebellar Purkinje cells are really a nice model. First, because it's the sole output of the cerebellar cortex, it will, any defects in the connectivity on Purkinje cell will have a clear behavioral readout, in particular in terms of motor coordination and learning. Second, it's been involved in defects in this connectivity have been involved in neurodevelopmental disorders. As I said, this connectivity and physiology is really well detailed. And in particular, it receives two types of excitatory input, one from granule cells through the parallel fibers and one from inferior olivary neurons through the climbing fibers. And it's a perfect illustration of the type of synapse identity that I told you about at the beginning, because each Purkinje cell receives more than 150,000 synapses from granule cells that are made on the distal dendritic spines, while each Purkinje cell in the adult receives a single climbing fiber from, at least in mouse, in most cases, that will form a few hundred synapses on the thick proximal dendrites. And we know that these two inputs are in competition for their territory of innervation on the Purkinje cell. If you remove granule cells, the climbing fibers take over and vice versa. So specific subcellular territory, very different numbers of synaptic contacts. And also, as you well know, the physiology of each one of these synapse type is very different, with inferior olivary neuron stimulation, climbing fiber stimulation giving an all or none response in Purkinje cell, while if you stimulate parallel fibers, you get a graded response depending on the stimulus and intensity. So a perfect example of the type of precise connectivity that we have in the brain. And furthermore, this network is genetically accessible in the mouse. We have drivers that allow us to express any transgene of interest specifically in each one of these neuronal population, making this network. So over the years, what we have done is to find new signaling pathways that are important for the formation and function of those synapses. And in particular, what we have identified is the role of complement-related proteins, both for synapse formation in this circuit and for synapse plasticity. So I'm not going to go, I'm not going to tell you about our study that looked at the role of SUSD4, for example, in the regulation of long-term depression and long-term potentiation in this circuit. I'm going to focus on another class of proteins that we have found to be important for synapse formation and synapse identity in this circuit. And those are C1q related proteins. You might be aware of the work of the Yuzaki and Mishina labs that a while ago now showed that the secretion of the cerebellin 1 protein by parallel fibers and the formation of the tripartite complex between presynaptic neurexin and postsynaptic GLUD2 receptors are essential for the formation and maintenance of this particular synapse. During our studies, we identified a new postsynaptic receptor, the adhesion GPCR brain angiogenesis inhibitor 3, and we showed that this receptor was essential for the formation of both types of excitatory synapses on Purkinje cells. And what we also showed is that the secretion of another C1q related protein, C1QL1, by the climbing fibers and its binding to BY3 is essential specifically for the formation of the climbing fiber to Purkinje cell synapse. So what this work suggested, together with the work of the Yuzaki and Mishina lab, was that a specific protein from the C1q related family was secreted by each excitatory input and was essential for the identity, the formation and the identity of these two synapse types on their common target, the Purkinje cell. However, what we also showed is that these proteins were not enough because when we removed them, only part of the synapses were removed, and you still had synapses that were made by parallel fibers in one case or by climbing fibers in another case. So what we decided to do was to actually embark on a project to kind of get the complete presynaptic code. What were the other molecules that were important for coding the identity of these two synapses made on Purkinje cells? And this work was done by two extremely talented scientists in my team, Maëla Paul, who was a PhD student, and Séverine Siguayo, and Maëla has now moved on to be a postdoc at Columbia, and Séverine Siguayo, a research scientist in the lab. And these two excellent scientists teamed together to really do a huge project that will be the main focus of my talk today. So the idea we had at the beginning was to say, okay, if we want to have the complete presynaptic code, what we need to do is to get all the complete gene expression profile of the granule cells, the complete gene expression profile of inferior liver neurons, compare those two profiles, and identify the specific genes for each input that code for membrane and secreted proteins, amongst which we will have the proteins that are important for the presynaptic code. We did that using the backtrap technology that was developed in particular in the Heinz lab at Rockefeller, and then using comparative bioinformatics. I'm not going to go into the details of this, but we can talk about it at the end of the talk if you want. This work led to the identification indeed of the two specific gene expression profiles for these two inputs and for the specific genes that coded for membrane and secreted proteins. And what you see is that we have a set of specific membrane and secreted proteins expressed by each input, which is bigger for inferior liver neurons, suggesting that these neurons are more complex than granule cells in terms of their surfaceome, in terms of the sets of genes coding for membrane and secreted proteins in these neurons. This has interesting implications. We can talk about it also at the end again. But what was interesting for us is that amongst those 74 and 250 genes, we should have those that code for the presynaptic molecular code for these two neurons. But these are a lot of genes. So in order to reduce the number of candidate genes, we took advantage of the knowledge that we have about the developmental sequence in this structure and in this circuit. So at birth in mouse, Purkinje cell receive several climbing fibers from several inferior liver neurons that make contacts. But then during the first couple of weeks, the external granule cell layer develops and starts to generate the developing and maturing granule cells that migrate along Berman glia to settle and form the internal granular layer underneath the Purkinje cell layer. And at the same time, what we have is the development of the dendritic arborization of Purkinje cells and the formation of the synapses with granule cells. So during this period, we have both the formation of granule cell synapses, the parallel fiber Purkinje cell synapses. And at the same time, we have the translocation of the climbing fibers on the proximal dendrites that also make synapses on these growing dendrites. So we have simultaneously these two synaptogenesis processes that are happening. But you've seen that these two processes don't happen exactly at the same time, and this is reflected in the pattern of expression of the two genes that I told you we know define these two synaptic types. C1QL1 starts to be expressed very early on, starting with the first contacts of the climbing fiber on Purkinje cells and peak around P14, whereas cerebellin 1, which is a marker for the parallel fibers, start to be expressed later on when we start to have the production of the granule cells and their maturation, and peaks later on at around P21, where you have most of the climbing, the parallel fiber synapses that are formed. So because we have these two patterns of expression with different peaks for the two markers, we decided to take advantage of this and perform high throughput expression analysis of all our candidate genes and compare them to these two patterns of expression. The idea, and you see here the result, is that then we can do unsupervised clustering to find the genes that have exactly the same pattern of expression in the cerebellum, like cerebellin 1, or in the brainstem, those that have the same pattern of expression as C1QL1. And you can see here that indeed what we had was one wave of expression of cerebellin 1, like cerebellin 1, of genes that are peaking exactly at the same time in the cerebellum. So this showed that granule cells, basically they express their surfaceome exactly at the same time as cerebellin 1 in one wave. In the brainstem, what we had was two waves of expression, one that was highly expressed during early development, and then these genes were decreasing in their expression, and another one which had a wave of expression later on, like C1QL1, with many genes that increased their expression at the same time. So for us, what was interesting were all the genes that were expressed exactly like C1QL1. So again, this led to a lot of genes that were expressed at the exactly with the same pattern as C1QL1. So we needed to refine a bit more our list of candidate genes. And what we had to do was database curation to select the genes that were highly expressed, those that had a structure that was of interest to us, so in particular adhesion modules. And then we reasoned also that for genes to be involved in synaptogenesis between climbing fibers and Purkinje cells, what we had to do was to identify those that had the single cell were exactly correlated with C1QL1. And this we did by single molecule FISH. Indeed, this allows us to do quantitation at the single cell level of the expression of our gene of interest and correlated to C1QL1. And altogether, all this work, which was really huge, allowed us to reduce the number of candidate genes that are involved in the formation of synapses between climbing fibers and Purkinje cells to about 3 in addition to C1QL1. And those molecules were 1 transmembrane protein, NRCAM, and 2 secretin molecules, SERTAC1 and LGI2. And these 3 molecules, we had to do a lot of work to actually identify whether they were localized at the climbing fiber presynaptic bouton. And indeed, what we were able to do by using various technologies, either CRISPR-Cas9 genome editing to induce an HA, to introduce an HA tag in the NRCAM locus, or finding different antibodies with different labeling protocols. It was really a huge amount of time to find the right ones. But you can see that for the 3 candidates, we were able to find to identify their endogenous localization. And in these cases, we could find them to be at least partially localized, co-localized with VGLU2, which is a presynaptic marker of the climbing fiber bouton, and to co-localize along the proximal dendrites of Purkinje cells where the climbing fibers are. So, for the climbing fiber Purkinje cell synapses, we found a combination of 4 molecules that have exactly the same pattern of expression during development, and that are good candidates for together being the presynaptic molecular code. To demonstrate this, though, as I said before, we need to see whether they are instructive. And for this, what we did was to develop a way to perform inferior olivary neuron specific loss of function at birth, and then analyze the consequences of this loss of function after a few weeks, when normally the synapses between the climbing fibers that you can see here in green are done and can be labeled by the VGLU2 puncta along the Purkinje cell dendrites that are visualized here by an anti-calbinding immunolabeling. So to do this, what we did was to combine viral injection strategies and CRISPR-Cas9. I'll spare you the details. And at the end, using our quantification strategy, what we were able to show is that NRCAM, this transmembrane adhesion protein, was actually inhibiting synapse formation during development when expressed in climbing fibers, while SERTAC1 and LGI2, 2 secreted molecules, when had exactly the same role as C1QL1, which is to promote synaptogenesis between climbing fibers and Purkinje cells. The next question was to address whether these 3 molecules had redundant function. And because we have this CRISPR-Cas9 technology, it's quite easy to do. And we address that by doing multiple loss of function during development. So here you can see that C1QL1 loss of function on its own leads to a reduction in about 30% in the number of synapses made on Purkinje cells. And what we did then was to add SERTAC1 loss of function, and that then we had an additive 23% decrease in the number of synapses made on Purkinje cells. And finally, when we did the triple knockout, we had an additive 18% decrease in the number of synapses. So overall, what this result shows is that these 3 molecules are not redundant in their function during climbing fiber Purkinje cell synaptogenesis. And I'll spare you the detail as well. We have some results on the volumes of the clusters that suggest that in some parameters, maybe of maturation of the synapses, they might have redundant function. So it's interesting because here we see that we have 3 different signaling pathways at this particular synapse that are connected to different types of receptors. So LGI2 is known to bind to ADAM receptors. C1QL1, I told you, binds to BI3 GPCR, and SERTAC1 is known to regulate RTN4R signaling. So these 3 molecules have different signaling pathways and could regulate different aspects of synapse maturation at this particular synapse, but they also have similar functions. And in particular, this is suggested also by the fact that they converge on a given signaling pathway, which is the one from the RTN4R receptor. Why am I saying this? I told you SERTAC1 binds to this particular receptor. LGI2 is also suggested to bind to this particular receptor, and C1 and BI3 is known to bind to this particular receptor. It has been shown by the Tom Sudoff lab recently. So overall, what I want you to retain from this information is that, as Perry suggested, we have a combination of molecule at a given synapse that is important for the synapse formation there, that this combination has partially redundant function, and unexpectedly, this is not only made by transsynaptic adhesion molecule, but this combination also relies on secreted molecules. So this was good, you know, this was one of the questions we wanted to address. Was Sperry right? Was there a combination of molecule for each synapse type? The answer is, at this stage, yes. But the surprise came when we looked earlier during development at our transcriptomic data, basically. What happened is that because we had this wave of early expression in inferior olivary neurons that I showed you earlier after our bioinformatic analysis, we went back to our data and we looked at all the genes that we had found that coded for the surfaceome in cranial cells and inferior olivary neurons. And we looked at the pattern of expression by taking all of them as one big set of genes. And we had the big surprise to identify that cerebellin 1, which I told you from the beginning is supposed to be a super specific marker of parallel fibers, is actually also expressed by inferior olivary neurons during the first postnatal week of development, and at exactly similar level as C1QL1. So what is cerebellin 1 doing in inferior olivary neurons, and is it important for climbing fiber Purkinje cell synapse formation? To address this question, we took advantage again of our CRISPR-Cas9 loss of function method, and we removed expression of cerebellin 1 from climbing fibers, from inferior olivary neurons and from climbing fibers. And we measured the formation of synapses on Purkinje cells. And as you can see, we could visualize in controls the formation of VGLU2 synapses on Purkinje cell somata by P7. And in cerebellin 1 loss of function, we found a significant decrease in the number of VGLU2 puncta, suggesting that climbing fibers couldn't make as many synapses during this first postnatal week on their target Purkinje cells if they didn't express cerebellin 1. When we looked later on at the formation of the climbing fibers, this time of their synapses on the Purkinje cell dendrites, again, we found a significant reduction in the number of synapses made by climbing fibers if they didn't express cerebellin 1. So cerebellin 1 is important for the formation of climbing fiber Purkinje cell synapses, both on the soma and on the dendrites. So this suggested that we had to revise basically the Sperry hypothesis, that it was only partially true. Indeed, in the adult, we have specific combination of molecules that code for the identity of each type of excitatory synapse made on Purkinje cells, but this was not the case initially. Initially, both types of synapses rely on a common molecular code that involves cerebellin 1, and the maturation of the code occurs during the first few weeks of development through a process during which inferior olivary neurons start to express an additional combination of proteins on top of the default code, while the granule cells remain in the default mode. And basically what this suggests is that the climbing fibers, through this active secretion of a new combination of protein, they kind of specify the pattern of connectivity on their target and maintain their identity in the face of the other inputs on Purkinje cells. The obvious question then is to try and understand what controls this switch in inferior olivary neurons. And for this, we had one usual suspect is neuronal activity, because neuronal activity changes a lot during these particular stages of development, and it has been involved in regulating, for example, multiple innovation by climbing fibers on Purkinje cells by many labs. It has also been suggested that neuronal activity in this network is even important in the adult by the Strata lab in experiments where they injected TTX in the adult rodent cerebellum and showed that this induced climbing fiber retraction. So we wanted to really understand if activity in inferior olivary neurons themselves is important for connectivity. And sorry, before that, also another suggestion that inferior olivary neuron activity itself is important for the proper formation of their synapses on the target came from a study that we did on the neonatal phencyclidine model of schizophrenia. So in this model, what you do is give a non-competitive NMDA receptor antagonist during the second postnatal week, phencyclidine, and this leads to long-term behavioral changes that are relevant for schizophrenia, changes in social behavior, working memory, and hyperactivity. What we did was to first test whether this PCP injection changed activity in the olivocerebellar network at these stages. And for this, we measured the level of CFOS expression. And indeed, both in the cerebellum and in the brainstem, injection of PCP increases CFOS, suggesting increased activity in the network. And then we looked at whether this change in activity could induce changes in gene expression patterns, in particular in those genes that code for membrane and secreted proteins in this network. And indeed, we had a change in the surfaceome, mostly in the cerebellum, when we injected PCP in the animal. So this suggested that maybe changes in activity in the cerebellum induced by PCP might have an effect on synapse formation. And to make a long story short, what we showed was that indeed PCP injection induces long-term changes in the number of climbing fiber Purkinje cell synapses. So this showed two things. First, that in a model of schizophrenia, something that was not so obvious at the time, but in a model of schizophrenia, you can have modification of the olivocerebellar network. And second, that maybe increased activity at this particular sensitive stage had a direct effect on synapse formation, and in particular on climbing fiber Purkinje cell synapse formation. To make a long story short, we tested whether this was the increased activity in the cerebellum that had this effect. We showed that this wasn't the case. You can see in the paper the chemogenetic experiments that we did to show that. But this overall suggested that indeed it's the activity in inferior olivary neurons that might be the switch that controls the formation of the mature climbing fibers synapses on Purkinje cell, and maybe of the molecular synaptic code. And to show this specifically, what we did was to reduce excitability of inferior olivary neurons, this time by expressing using viruses the channel KIR2.1 specifically in ION at birth, and looking at the consequences after 3 weeks. And indeed, what we saw was a big decrease in the number of synapses formed by climbing fibers who had reduced excitability during development, compared to the controls expressing the KIR2.1 mutant channel. The next question was whether this also changed the acquisition of the mature molecular synaptic code at the climbing fiber synapses. And for this, we combined this expression of KIR2.1 in IONs with a quantitative measurements of the expression of our genes of interest. So first we tested that in cortical cultures, and as you can see here, we showed that KIR2.1 expression not only decreased FOS as expected, but also decreased C1QL1 and LGI2, two of our candidate genes. And when we did this quantification in inferior olivary neurons, again, we showed that C1QL1 and LGI2 were decreased. Interestingly, only those two genes were regulated by activity, not SERTAC1 and not cerebellin-1, suggesting that it's not all the molecular synaptic code that is regulated by neuronal activity during postnatal development. So overall, what I've shown you today is a new model, a new framework to think about the way that neural circuits are built during development and how we acquire synapse diversity and specificity during development. What we suggest is that initially synapse formation is promiscuous. Different types of neurons that have a common target rely on the same molecular signaling pathway to form initially synapses, but then partly through neuronal activity dependent mechanism, there is a switch in the pattern of expression in one particular neuron that starts to express a unique combination of synaptic proteins that allows the specification of its identity in the face of the other inputs that are made on the same target neuron. This, we think, probably is relevant for other circuits in the brain, not only because of the stereotypic patterns of connectivity that I illustrated to you at the beginning, but also because many of the molecules that we identified are expressed in other networks. So I'll end here by thanking the people in the lab. I already told you that Maëla Paul and Séverine Siguayo were really instrumental in doing all this work, the facilities and our funding, and you for your attention. Thank you.
Fantastic talk, Professor Selimi. So I will go ahead and start us off with a question, and feel free others to raise your hand as questions arise. So start us off. Thinking back to Lackey and Rager's 2024 work and Bol and Tuzeyu's in 2018, there's this growing notion that there's something going on in the molecular layer associated with P-cell computation and activity. So my question is, well, I guess specifically MLI1s or stellate basket might participate in PFPC synapses, and MLI2s receive some kind of climbing fiber spillover. So were there any synaptic developmental differences or DEGs or anything of that nature related to maybe position in molecular layer or to molecular layer into neurons themselves?
That's a good question, and I think we still have to look exactly at how these molecular layer interneurons make their synapses on Purkinje cells. What is interesting is that there is a rather old study now from the Kano group, if I'm not mistaken, that shows that if you knock out the alpha-1 subunit of GABA receptors in Purkinje cells, interneurons will make synapses on spines that look like parallel fiber spines and that have the GLUD2 receptor, which is the receptor for cerebellin 1, which is the default molecular code. So this would suggest that even molecular layer interneurons share this default molecular code initially, but it's through activity that they kind of remove this default molecular pathway from their synapses and acquire a mature aspect and a mature code and a mature morphology. It would be interesting to kind of test this live, basically, to see whether we could see this evolution during development. Yeah. And in terms of their position in the molecular layer, this has to be really analyzed, I think. Yeah.
Wow, that is eye-opening. Next is Solomon. If you would unmute.
Hi, thanks so much. So if I understand correctly, the idea is that for the climbing fiber connectivity to the Purkinje cells, it depends on the activity of the climbing fiber. But what would determine then the Purkinje cell, the postsynaptic? Because I think you're arguing that the molecular layer develops after the climbing fiber.
Yeah, so you have a two-step process, and actually that what that means is that it reduces the problem for the brain, right? Because if you had to form the network the way it is at the end, then you really would need to have a lot of specific signaling right from the start, right? Here you reduce the problem because what you have is Purkinje cells. They have this delta-2 receptor pretty early on their soma, on their dendrite. And so you have this input that come and recognize this as their target, right? But then indeed, what you have is at the same time as you have the granule cells that are produced, the dendrites are growing, right? And those dendrites, they still have delta 2, so it's still the same molecular code, right? So at this stage, everything is mixed, and it's been shown even by serial EM that parallel fibers and climbing fibers at that stage, they share the territory. They're intertwined. But then you have to somehow make those neurons understand what is their territory. And so you have to have one of the neurons that says to the other one, you have to get out of here. This is where I'm supposed to be and not you. So it simplifies the problem. And to have this, it seems to be that it's the climbing fiber and the inferior olivary neurons that are active. So you have to change the code only in them, basically, and this is through this neuronal activity.
Okay, thanks. So if I may, in other words, when you have two competing climbing fibers, so is it the case that only one of them active? What happens if both of them are active? How do you win? Is it depending on the post?
Yeah. So that's very interesting because all the work that was done up to now, even at the neuromuscular junction, but it was also there are studies that suggest exactly the same thing in this particular system, is that you need to have differential activity to select one climbing fiber. And usually it's the strongest one that wins, right? If you have either all of them active at the same level or all of them inactive, then you maintain multiple innovation. So now it seems that it's not all or none in the sense that Christian Ansel has published a beautiful paper recently that's showing that multiple innovation might be more frequent than what we thought in the mouse, depending on where, in which lobule you are, in humans more in general. But so that indeed brings question as to how exactly this neuronal activity also controls the difference between the different climbing fibers coming from different olivary neurons, basically. But this adds another complexity to the system, right? Because this is the control of the final topography in the network. So you have a competition between the two inputs, which are granule cells and inferior olivary neurons. But even amongst inferior olivary neurons, you need to somehow regulate this. And this comes back to what I was saying at the beginning, is that the surfaceome of inferior olivary neurons is much more complex. And probably that has something to do with this topography as well. So subclasses of inferior olivary neurons probably expressing different patterns of molecules to control in an activity-dependent manner to control the topography. Yeah.
Thanks.
Any other questions from the group? All right. Well, thank you so much for joining us today, Professor Selimi.
Thank you for the invitation.
Of course.
Bye-bye.
Okay.
Ciao, ciao.